Okay, so what we're going to do now is just take a few minutes and explore the structure of DHODH and visualize the positions of the mutations that Paul's been telling you about. And we're going to use a program. There are a number of different programs for looking at structural data. This is one that I like, which is called Pymol. It's easy to work with. It's open source. A lot of it is written in Python, so you can sort of hack away at it if you know a little bit about computer programming. So what we're looking at right now is one representation of the data that comes out of one of these X-ray crystallography experiments. So what we have is each of the non-hydrogen atoms, so those are the heavy atoms. You can't see where the hydrogens are in X-ray crystallography, unfortunately, but you can see where the heavy atoms, so carbons and oxygens and nitrogens. So for each of those atoms that's visualized in the experiment, there's a vertex in this visualization, and there are lines connecting pairs of atoms that are bonded. And so, of course, what we're mainly seeing here are all of the lines connecting the atoms that are joined by bonds. And so we can then zoom in on a particular part of the protein. So, for instance, here is a particular here's a particular amino acid, an arginine, and we can look at that in a little bit thicker view. We can but here again, what we're seeing are the bonds essentially that make up that individual amino acid. So there are a lot of other ways of looking at the protein structure that can give us a bit more insight. I mean, when we spin this thing around, we just see lots and lots of lines and colors, and it's not the most intuitive representation. So what people, protein crystallographers and structural biologists have been doing for a while is figuring out different ways of representing a protein structure. So this data, which is a big long set of X, Y's, and Z's, in ways that are intuitive. And so, for example, they've come up with what are called cartoon representations. And so here's one. We can just, so this is showing what are this is now just showing the path of the backbone of the protein. So you guys probably know that a protein is a polymer, a linear polymer, composed of amino acids strung one to the next. And this is showing just the path of the backbone of that chain. So it's sort of like a long necklace that wraps around in space. And the protein, when you look at protein structures after they've solved the first 5 or 10, they realized that there were certain recurring patterns that they saw over and over again. And you can see them here. There are patterns. These are patterns are called secondary structure, the secondary structure of proteins. And here's one which is called an alpha helix, this little helical structure here. You can see a lot of our protein is made up of alpha helices. And then there are also these little arrows in here, which are beta strands. These are just different ways that the protein backbone likes to fold up in space as it travels through space. And they give coherence and some sort of order to the overall structure. In fact, you can kind of see, this is a really beautiful protein structure. You can see the way that the arrows, the beta strands here, form the middle. In fact, if you could see the way they're connected, it's almost suggestive of a barrel, and the strands are the staves of the barrel. And then we have the alpha helices around the outside. Okay, so that's a view in which we can get more sense for the way the chain moves as it goes through the protein. And in fact, we can color it. There are all sorts of ways we can fiddle with things. We can color it running from one end of the protein to the other, which is also kind of nice. So we can see that the protein starts over here in dark blue, and then we travel through the chain, and then into the barrel region here, and we go beta strand, alpha helix, beta strand, alpha helix, beta strand, alpha helix, all the way around to the red. So anyway, so those are just different ways of trying to make sense of this complex data set that we have in the form of a protein structure. Let's see. Another nice view is sort of the view that if you were a water molecule bouncing around the outside of the protein, the view that you might see, which is called a surface representation. So let's just get rid of the cartoons, show the surface, and let's color it in a more naturalistic kind of representation where we have carbons gray, nitrogen's blue, oxygen's red. Okay, so here we're looking at sort of the outside of the protein, if you will. This is the part of the protein that a water molecule bouncing around can actually feel. And one other thing that's important about this enzyme is that it has, that it's an enzyme, so it operates on small molecules, on ligands, and we can actually show them too. So this is the fun thing about going into a protein structure, is trying to see all the different moving parts. So let's show these cofactors or ligands, they're called. So let's show them as sticks. So these would be the things that the enzyme is operating on. And to see, they're of course kind of hidden away inside the protein. So if we can maybe try and cut away here a little bit and see if we can find them. So this is just kind of a fun way of looking inside the protein. And now, in fact, we can see, so this protein is an enzyme. It changes one molecule into another. And we can see the molecules here bound inside the enzyme that are being changed in the course of the reaction. So this is one of them here. This is a flavin mononucleotide. And then we have orotate up here. And you can also see that this protein has kind of these hollow areas inside here. So these are areas where if you were a water molecule, you could potentially probe inside the protein. The vast majority of proteins are not accessible to water. They form sort of a greasy solvent excluded ball oftentimes. But there are these little tunnels, and in enzymes these tunnels are really important. And so, for example, the tunnel that we can kind of see here in cutaway view is the tunnel by which one of the important substrates of the enzyme can access the active site. So these particular structures here were solved because scientists wanted to understand how chemotherapy agents that target this enzyme actually work. And so we can show them. Those chemotherapy agents were resolved as well as the protein and its natural substrates. And so if we show them here, so yeah, so here's one of them here. And what's really neat about the structure is that you can see that where this small molecule, this drug, this chemotherapy agent binds, is right in the tunnel, which is an important access point for one of the substrates of the enzyme. So mechanistically, it becomes pretty clear how this chemotherapeutic agent is blocking the activity. And again, I should say this is not my area of expertise at all. But when you look at these structures, you can make up stories that are often not so far from the truth. Okay, so again, that's the surface representation, which lets us see these interesting tunnels and voids inside the protein and see where the active site of the molecule is, and also where this particular inhibitor is binding. Okay, so let's go now to a view that helps us understand something about the mutation that Paul is interested in. Let's go ahead and get rid of this surface, and maybe show again the cartoon. Now let's zoom out so we have everything present. And so there again is our protein, and now I've got the substrates and inhibitor present in the cartoon view, so we can sort of see where they sit. This is a really common motif that these barrel enzymes bind their substrates up at one end of the barrel. So we've got all these little arrows here, and they're all pointing up toward the active site up there. So where are the mutations? So we can, I defined those earlier, and we can show them in another representation here. In fact, we can also color them a little bit differently, so maybe they'll stand out. Okay, so those are the two sites of mutation. They're both glycines in the wild-type structure. And just to get a sense of the relationship between the linear sequence and the three-dimensional structure. So here, for example, is now shown at the top the one-dimensional primary amino acid sequence of the enzyme, and we're seeing it above the three-dimensional structure. And what's neat about this and all protein viewers is you can take a stretch in one dimension and see where it falls in the path of the chain in 3 dimensions. And so for instance, we can see then in the case of these mutations, the regions of the structure where they fall, and we can begin to try and come up with stories about what they might be affecting. And so let's see. So again, there are 2 mutations of interest. They're both mutations from glycine to another amino acid. Glycine is a really special amino acid. It's the smallest of the building blocks of proteins. And so it's often found at important places where the protein chain makes a turn, and that's certainly the case here in the case of this mutation at position 153 or 152, depending on your numbering system. Here the protein is making kind of a tight kink here, and the backbone conformation is one that really only a glycine can be happy in. So I think that's one of the reasons that this glycine to arginine mutation is destabilizing. One thing we can do, let's see, let's get a little bit more sense of what's going on right around the glycine. So we can within all these programs, you can define particular regions of the protein that you're interested in. So let's select the neighbors, and I guess what's really striking to me as a structural biologist is that it's really packed very tightly around that glycine. So there's not a lot of space for other stuff to fit there. And in particular, if we were to mutate that glycine to an amino acid that was bigger, which would be pretty much any mutation, but in particular the mutation that we're interested in here, there's just not space to fit everything. And so, for example, we can see, we went ahead and modeled in a mutation to an arginine, and now I've gone and shown that. Maybe I should, let's turn that a different color. So this is the mutation in question, modeled by keeping the backbone of the protein completely fixed, which is just the simplest way to model any kind of a change in the protein structure and a good first approximation. And what we can see is that this arginine now is kind of bumping out. It's let's see. So let's show. So I'm used to looking at these, and what I know is that these are like way too close for comfort. But we can show them in another representation that'll make that even more clear. If we show, for instance, spheres of, let's see, this neighbors set, right? Let's make sure we don't have waters on. And then we can also show in spheres that mutated arginine. So let's just select that guy. So this is now what the spheres representation is showing, you know, at spheres that are centered at the XYZ coordinate of each of the atoms, and whose radius is roughly the radius where they like to pack up against one another with the spheres just sort of gently touching. And so this this amino acid here, which is right next to the glycine, which is a proline in the neighboring loop, its spheres are overlapping with the spheres of the arginine, which is just sort of a kind of silly way of saying that they're bumping into each other, and there would be a terrible, terrible energy if this structure actually occurred. So most likely what happens when that arginine mutant is present in the folding of the protein, one of these two loops, and probably both, have to move a little bit to accommodate this change in the size of the glycine residue. And that most likely perturbs the stability and probably also some aspects of the function of the protein. Let's see. So that's the one mutant. The other mutant is down here. It's not as catastrophic, you might guess, by the amino acid change. It's a glycine to alanine, and so this is the mutated side chain. But here again, it's very tightly packed in the neighborhood of the glycine. So I think there too, we'd expect that the backbone is going to be forced to move a little bit to make room for the mutated side chain, and that there's probably some kind of cost in the stability of the protein. Okay, so let's see. We modeled the mutants. Let's go ahead and get rid of the spheres. We talked about the overall backbone, the cofactors. Yeah, maybe that's those are the main points that we want to look at here on the screen. Okay, so